Polypeptide is a chain of amino acids. These amino acids are linked through a covalent bond. These chains of amino acids go into proteins. Proteins are required for the structure, function, and are responsible for the regulation of the body's organs and tissues. In the polypeptide synthesis is the process of making a polypeptide. Polypeptide synthesis occurs within the nucleus. During polypeptide synthesis, the two main tasks occur. These are called transcription and translation. Transcription occurs within the nucleus, alongside the DNA. DNA, or deoxyribonucleic acid, is a self-replicating material that is present in all living things that stores information that creates proteins. DNA is a double helix, meaning it is made up of two strands. These two ladder-like structures are made up of nucleotides. A nucleotide is made up of a sugar molecule located either in the ribose in the RNA or deoxyribose in DNA, and is attached to a nitrogenous base. The nucleotide is made up of three components: a nitrogenous base, a phosphate group, and a deoxyribose. These deoxyribose and phosphate form a structure to uphold the back of the DNA connected to our four nitrogenous bases. These are adenine, guanine, thymine, and cytosine. These are attached to the backbone or the deoxyribose through a chemical bond called a glycosidic bond. Polypeptide synthesis begins with an enzyme called helicase. Helicase unwinds this chain of amino acids. One of the strands of amino acids will perform the DNA replication. These amino acids come with a complementary partner. Adenine goes with thymine, guanine goes with cytosine. However, within the RNA, if adenine is placed, uracil will replace the thymine. Shortly after, the RNA polymerase will join back these nucleotides together, forming the mRNA. This mRNA then travels to the ribosome, which marks the beginning of translation. Within the ribosome, the tRNA carries various amino acids. This is then linked back to the mRNA, where the tRNA and mRNA will join together. The mRNA will hold codons and the tRNA will hold anticodons. This continues as the next anticodon connects to the codon, making a peptide bond through amino acids. This continues as the anticodon goes but leaves the amino acid with the next anticodon. This will continue until a stop codon, or one that doesn't code for a specific amino acid, is reached. This then creates a polypeptide. This makes up the structure of polypeptide synthesis, although with this amino acid chain or polypeptides come what are known as point mutations. Point mutations are the changes in a gene structure of the gene. There are a few types of these changes that happen with a mutation: 1. Deletion, 2. Insertion, or 3. Substitution. An example of a mutation is sickle cell. Sickle cell is when the red blood cells aren't a normal shape, meaning they cannot transport sufficient amounts of blood. Within an individual with sickle cell anemia genes, the mutation occurs within substitution. Within this mutation, a single base is replaced in the DNA coding for hemoglobin. This hemoglobin is a multi-protein complex responsible for the shape of blood cells. This means that through the sickle cell anemia mutation, the gene is substituted from a glutamic acid to a valine amino acid. Within the amino acid chain shown, you can see that the fifth amino acid changes to the amino acid called valine. As you can see, the amino acid sequence will then substitute CAC to CAT, making the nRNA GUA, meaning the substitution goes from histidine to valine. Therefore, if this human individual cell, they would have sickle cell anemia. This means that sickle cell anemia can be harmful to the continuation of species through the change in amino acid sequence that leads to the disease and its side effects.